Hi everyone, this is Julie Hoggett, Senior Principal with inThought Research. With ASCO 2020 less than a week away, I'll be providing inThought's preview of the key presentations from oncology's biggest annual conference. Key data sets will be presented on Roche's anti-TIGIT antibody tiragolumab, Allogene's allogeneic CAR-T ALLO-501, and Karyopharm's Xpovio in second-line or greater multiple myeloma. Likely the most anticipated presentation of ASCO 2020 comes from Roche's phase 2 CITYSCAPE trial, assessing the combination of their anti-TIGIT antibody tiragolumab with Tecentriq in first-line non-small cell lung cancer. This work will provide the first full glimpse of anti-TIGIT efficacy and safety, which has been billed as the next major checkpoint inhibitor. In PD-L1 high expressor patients, tiragolumab and Tecentriq combination demonstrated an objective response rate of 55.2%. As far as chemo-free options go in non-small cell lung cancer, the data looks promising. Keytruda monotherapy, which is FDA approved in first-line non-small cell lung cancer, has demonstrated a 27% objective response rate with a 5.4-month median progression-free survival, or PFS, in PD-L1 positive low expressors, and a 39% objective response rate and 7.1-month median PFS in PD-L1 positive high expressors. Based off this primary analysis, the tiragolumab Tecentriq combination has thus far underperformed in the PD-L1 positive low subset, but outperformed Keytruda monotherapy for PD-L1 positive high expressors. If these results can be replicated in a phase 3 study, the tiragolumab Tecentriq combination may prove to have the upper hand in the PD-L1 positive high subset. Further, the 55% objective response rate is on par with currently approved PD-1 axis inhibitor plus chemotherapy combinations that are approved in the first-line non-small cell lung cancer setting, where objective response rates range from 43 to 58%. Another closely watched presentation will be on Allogene's allogeneic CAR-T ALLO-501 in relapsed and refractory non-Hodgkin lymphoma patients. ALLO-501 is differentiated from currently approved autologous CD19 CAR-T therapies, Yescarta and Kymriah, as it's allogeneic or off-the-shelf. Further, this trial is also combined with ALLO-647, a CD52 targeting antibody used for lymphodepletion in an attempt to avoid the harsh conditioning regimens typically associated with CAR-T therapies. ALLO-501 is produced from banked healthy donor cells and is edited to inactivate the endogenous T-cell receptor by a TALEN inactivation to avoid the prospect of graft versus host disease, or GVHD. This strategy is aimed at overcoming the manufacturing shortcomings that are associated with autologous cell therapies. The two predominant concerns of allogeneic products are avoiding host rejection and GVHD. GVHD has yet to be observed for ALLO-501, but with a median follow-up of only 2.7 months, we await more data on persistence. Further, the absence of serious cytokine release syndrome and neurotoxicity to date, a side effect that has limited Yescarta and Kymriah, is notable. Although the current sample size is small, ALLO-501's efficacy with an objective response rate of 78% compares favorably to Yescarta, which was 72%, and Kymriah, which was 50%, in a cross-trial comparison. Updated results are anticipated at ASCO 2020, but this proof of concept snippet has many excited. The last presentation we'll preview is Karyopharm Therapeutics Phase 3 Boston study of Xpovio in combination with Velcade and dexamethasone, also known as SVD, in second-line or greater multiple myeloma. These trial results were submitted to the FDA on May 20th and aim to convert Xpovio's accelerated approval to a full approval while moving Xpovio to earlier lines of therapy. The second-line setting is a strong opportunity for Xpovio, as no regimen is dominating the market. SVD's safety profile will be closely monitored during this presentation, with an emphasis on peripheral neuropathy rates. 3 other presentations we'll be keeping an eye on include data on Autolus's AUTO3, CD19, and CD22 targeting CAR-T and diffuse large B-cell lymphoma. There's also 2-year follow-up data from Merck and GSK's TGF-beta and PD-L1 targeting bintrafusp alpha in second-line non-small cell lung cancer, and patient responses after treatment with Trillium Therapeutics' CD47 inhibitor, TTI-662 in lymphoma. Thanks for watching, and keep following us on Twitter and LinkedIn for more conference previews.